Good morning, good evening, hello to where you sit and listen. I hope you're fine, well. This week is an open medical cannabis topic, and this is going to be a legal show, and it promotes the law and it promotes safe uses of medical cannabis and safe growing practices as well. I don't promote anything that's undue to the law or undue to the community, and I like to grow things healthily, and that's the only way that I'll speak. So if you want to do illegal things or anything like that, this isn't the stream for you. See ya. But to those others, I'll say g'day. So hello to Dave, 1969. I hope you're well. Thanks, mate. Jeff Papalia. G'day, Jeff. And Vinny, how you going, Vin? Nice to see you. It's a open stream, open topic today. So if you've got anything that you want to start off on, please let me know, because I haven't got anything that's planned. Otherwise, I'll just be starting into my stuff my plant soil science and microbiology stuff that's related to medicinal cannabis. So please ask questions, and I'll see what slides I can find related to whatever you want to find. I'll give you a bit, because this has got a bit of a lag for questions, and then if there's no questions, I'll start and pull up some slides. So I think already I'm just going to try and have a bit of a squeeze for something just in case, because things are moving a smidgen slow. So what can today? Plant science. It'd be good to talk about microbes. I haven't talked about microbes for a bit. Oh, well, plant science. What's good, cool stuff in here? Hormones is always a good topic. A good botanist is supposed to be able to manipulate any pathway through hormones. All right, I'll just check chat again if there's nothing on that. I might do a little hormone discussion. Where is it? There we go. G'day, Supreme Grape. How you going? Why didn't it show up there? Bush Rangers Journey. How you going, Bush Rangers Journey? Today is an open medical cannabis topic, so if you've got any questions, please feel free to ask, and that'll start the show. Otherwise, I'm just going to start sharing some maybe plant hormones today. That's what I sort of tickled my fancy just there. How you going, Sunshine? Sunshine Rainbows. Thank you for support. Oh yeah, that little money underneath. That's for the support button. I appreciate those who have a lot. All right. Well, without good hormonal structure, you're not going to get an end result that you want. The yield might be lowered. There might be other things that will interact and slow things up, the processes and the pathways. Just like a human, if it's given hormones, it will, in the right medical environment, it will boost the area that it's trying to fix. I remember having cortisone in my knee, for instance, and that used to get help and used to boost the pathways that link up for that. So instead of it making the hormones that it wants, it's given it, bottle fed, and I'm talking without showing stuff. I'm just, all right, I'll just run through these. These are the basics. Where is the, back to here. This is no one's, yep, there's no questions. So share screen. We're going to get into hormones today. Hide bookmark. Where is it? Entire screen will do. Everybody can see what is going on. So here, so these are the three main. There's about 8 different pathways of growth stages in medicinal cannabis, but for here it's stated as 3. So at the start, when you're germinating or cloning, you'd want a lot of gibberellic acid and then a little bit more cytokinins. Cytokinins promotes root division. Cell division. It elongates the cells. And then auxin also helps as it gets a little bit further, and that's the blend that's going to mix, and that's going to give you your roots. So this will produce shoots, and this will be in for the roots. Majority, they do other things. And what's the other ones? Cytokines, auxins, gibberellic acid. Okay, well, that should have been back here. Oh, it is back here. Gibberellic acid helps in the pathway of seedlings when it's looking for the inside of the seed. When it's going to germinate, it first needs three things: your water, oxygen, and temperature. And then it'll activate the amylase, which breaks down to start to produce the breakdowns for the sugars, and then that will form that will activate the pathway for the gibberellic acid. So that's another thing that helps for germination. And in the grow, well, gibberellic acid helps in a lot of pathways. It's pretty good. But you can always give too much, remember. A lot of these things are so minimal amounts. So I've found out in tissue culture. I'm just going to make sure that I'm sharing. I had big problems last one. I'm speaking, I'm sharing. I hope everything's going well. All right. And you get through the grow and ethylene starts to form. And that's when it's in the senescent stage. And you don't want ethylene because it's going to promote senescence, and it'll tell everything else to finish. I made a pretty detailed video on how cannabis works and how senescence works in cannabis, different ways you can manipulate it through hormones and through different, like auxin, for instance. There's some cytokinins you can rub on the leaves. There's some other different pathways that you can activate through to eliminate the senescence. So that's a pretty good video if you're into that type of thing, want to extend your crop, or if you want your crop to finish early. There is some ways to do it, indoors and outdoors too. Abscisic acid forms also when it's going through senescent, and that's the ABA promotes deaeration. It drops off the leaves when it promotes through senescence, and it's not good. It's really good finishing, of course, as you can see here. Anyway, that's just one chart. There's so many charts to go through. That's a sort of another looking chart. Actually, I'll see what's happening written in here just in case. Let's go back to this one. G'day, spring grape. How you going? Yeah. Lene, nice to see you, Lene. Good morning to you. I just watched some person make a video saying how bicarb soda is the best fertilizer. Well, it's not. I can tell you bicarb soda is full of bicarbonates, and that's OH, what's that, the carboxylate? And that's, if you want to get into the pH, that's the thing that raises the pH. So if you want your hydroxyls, which is your protons, which is your H pluses, that's lowers your pH, and your OH raises it. So it's just worked good with him because he's probably had an acidic base. I'll just scroll down. Oh, I'm already down. Very good. All right, back to hormones. It's not hormones. This one is. All right, so I suppose I'll go through. See, there's 10 hormones. There's 5 basics, and there's 5 that are not basic. So you've got your auxin, cytokinins, gibberellins, abscisic acid, and ethylene, and then it goes into your salicylic acid, your jasmonates with your jasmonic acid, brassinosteroids, strigololactones, and florigen. These other ones are fantastic. I like playing around with salicylic acid and jasmonic acid. They're good at promoting stresses in plants. Brassino is good for vegetative growth. Strigolose, I can't remember. Should I go through all this? I bet she's going to be huge in here. Well, I don't even know if you guys want to see this. I'll just go through here, see what's in here. Cytokines, auxins. Auxins help in phototropism because what they do, they pull the auxins to the sides of the cells, and if you stain it, you can see the sides, it stretches out the cells here. So these sides here are stretched, so it elongates it and makes it go into phototropism, which means it moves. Synthesizes stem tips. It travels downwards. It has a polar flow. It only promotes in the tips, and it gets traveled to the roots. So if you go and top your plants here, you've got to stunt for about 2 days because there's no auxin, because you want to maintain auxin maxima, it's called, when the amount of hormones in your shoots is producing its auxin at its best, and it's going to give it the best hormone production. If removed stem tip, plant loses ability to grow towards light. Yeah, it's only for a few days until it it branches out of its other nodes. Gibberellins require to germinate and promote elongation in internodes. Yes, if you've got a dwarfing, like one of my varieties has a DWF death bubba. It's got a dominant DWF gene, which means it's a dwarfing gene. So to get around that, I've gone and give it little bits of gibberellic acid, which is a G3, which is the type. There's about 100 different types of gibberellic acid. And yeah, I'll give it the G3. And it's like tiny, tiny amounts. I made a mistake of giving it too much. It elongated it too much, stretched too much. And I got I think a bacterial infection, because when you stretch it too much, the cell walls shrink. They stretch because they've got to pull from somewhere. So they're still maintained its same original size, but they're stretched now. So like a rubber band, when you stretch it, it goes really thin. And for piercing or cell-sucking insects, they can easily penetrate through that. So I would have had some sort of vector infection passed through. Cytokinins, yeah, countersenescence. You can rub it on the leaves if you want your plants to go longer. Roots and shoots. It works with the roots and shoots with equal, very good with abscisic, with auxins. Works with ethylene to cause abscission. That's the word I was looking for before, abscission. That's what abscisic acid does. It abscises the leaves. It makes them die at the end so it can fall off. Abscission. That's just where it falls off. Root apical meristem. What's that? Okay, very good. That's the tips of the roots. That's where the auxin flows down to. And if you put a stain into here, you can get it'll light up, showing, wow, look at all the auxin in there. Abscisic acid. Yes, it singles dehydration. So if you want to have a drought-resistant variety, your cultivars are sick of wilting all the time and it's very hot and dry where you are, you can breed with abscisic acid to have that outcome. It makes leaves close their stomata to prevent water loss through phototranspiration. So that's the chlorophyll there in the grana that are closing. Actually, it's potassium ions that actually close it as well. They react pretty well with that, the stomata. Ethylene ripens fruit. Oh yes, it does. So if you want your plants to finish early, put more ethylene in, just like they do with bananas. They build big rooms with ethylene, pump ethylene in for bananas. So I pick them really green, and then it'll get to the shop or close to the shop, and I'll put them in these rooms, and I'll just ripen them very fast, depends how much they want. Ethylene also promotes rapid growth of cell division. In germinating plants, it prevents leaf expansion. Actually, in germinating plants, it gives a triple effect, it's called, and that's it has a shortened hypocotyl, so it'll be short, it'll be thick, and the root, the radical, will have a J hook in it. And that means that you've got too much ethylene in your area where it's germinating. And it's also very good to use for dormancy. The ethylene also allows shoots to dig upwards through the soil until it reaches light. I haven't tested that one. What's this? Inhibitor. Oh, you don't want an inhibitor? So to produce ATP, adenine triphosphate, that's the cell currency, that's the things in us humans too that produce energy. So that's, if you want more metabolism, this is where it starts, at ATP. And it turns out carbon monoxide is a blocker of ATP. So if you've got carbon monoxide comes out of exhausts and from cooking, so if you've got those around, that's what might be a reason why you're getting reduced growth. What's this one? Phytohormones, old timers and newcomers. Oh, yeah, that was those ones that I explained, the original ones and the new ones. Plant cell teaching tools. Phytohormones regulate all stages of plant cycle growth. Yes. One of my assignments was to document that. So I went and documented it in Cannabis. And I've got this really rad A4 page full of how to do this and that with the different hormones and what it does, what effects it does on the plant and that sort of stuff. So beneficial, mate. They're really, really good to work with. A good botanist can manipulate any growth stage of a plant through hormones. So I thought, oh, this sounds interesting. So I, looked a lot into it. That's basically what that page says. It manipulates everything. Every pathway needs hormones to promote and to activate it and to get it kicked into that and knocked out of that. Here's some of the roles of the hormones. So the hormonal control for vegetative growth is quite a lot of them. The hormonal control for reproduction, ethylene and abscisic acid, because that will promote, if your plant's going into, it throws out bananas, if it herms, throws out anthers, that means that it's starting to reproduce itself. So for that to be triggered, its pathway was stimulated by a abiotic or biotic stress. And ethylene and abscisic acid is one of those abiotic stresses that can happen. A biotic stress is bio, meaning life. So it has to have something life. So this is an abiotic stress. Hormonal response to stress, yes. Salicylic acid and jasmonic acid are other two ones. Same thing for the stress, if you really want to play around with it. This is germination of a seed. This shows you your abscisic acid stopping the pathway. If there's too much abscisic acid, it won't promote it. And it shows you that gibberellic acid, it increases it because of its pathways I explained earlier. So light plus gibberellic acid helps rupture the tester, and the tester is the seed coat on the outside of it. So if it's dormant, you can actually germinate a medical cannabis without light, and it's an elitiophile, elitifiolate. It means that it hasn't picked up any elitiolate, something like that. Not pronunciation. It picks up the photosynthesis normally for a plant, and that will activate the chlorophyll pigment. And because it hasn't had any light, it won't activate that. So you'll get leaves that are just white or just yellow. They haven't got any chlorophyll inside of them. And endoplasmic, okay, ABA, yes, that's what. Some of them, ABA, see how it's negative, so it stops it, and then ethylene promotes it to plus. So if you get too much in there and then it germinates, you'll get your triple effect from ethylene, if you know. So that's a good indicator for ethylene to know if you've got too much in there. If you're cracking dormancy, radical, that's enough for that one. That's pretty cool. Oxygen ratio. That's tissue culture. Well, it is involved in tissue culture and medical cannabis, and tissue culture, actually, I should do another topic on that. It's bloody excellent. It's really fun too. So easy, takes up little space. You just need like a tote. Just turn a tote on the side of a big plastic container. And then, make sure it's very, very sterile. And even though you don't have to have it very sterile, because you'll find that some of your techniques will still work. And by doing cloning, cloning, that's a form of tissue culture because you're trying to produce roots. Just this way, you can manipulate it just by using auxin cytokinins, by using, if you want to get all roots coming out or all shoots, or if you want, you put a good blend of both of them, and it just promotes a good callus. And that's like that thing, this blob in the middle. And then from that, you'll cut a bit off to a subculture. And then you go, what do I want from this? Oh, I want plantlets. So you go and put more cytokinins and you'll get you more plantlets. Trichome formation, yes. So if you want more trichome formation, it's involved in hormones as well. I spoke about a few weeks ago. So these are the genes you want to activate to get more trichomes. The control of the QC center. So the QC center is right in the middle of the root tip, quiescence center, and that's a area that has no, it's a stem cell niche because it has no designated pathway yet. So it keeps reproducing and forms. Actually, does it reproduce? No, it stays the same and it reproduces all around it. And all these ones are cells around it. They're the ones that get designated pathways into something. So the WOX5 gene, that's an auxin one. They're all involved in these different formations of what is going to get promoted around the QC. So it depends what depends what hormones come in as to what's going to, it's going to give it its little tick. All right, you can go and produce roots. You can go and produce shoots. We'll start and do this. So you can get some real funky things happening. That's plant biology. What's this one? There are a few different ways to fiddle with it. Okay. Cannabis chemistry. That's like an overall thing. You don't need to talk about that. It just works. 150 cannabinoids. Wow. What's this one? Xylem phloem. So this is to do with the shoot development from different auxins and their pathways. Do I want to explain it all? Do you want me to explain it all? Actually, I'm going to stop sharing and see if there's any questions. Far out. Sorry about that. Getting a bit carried away here. I'll scroll up a fair bit by the look of it. Gee whiz. There we go. G'day. See you every day. How you going, mate? Nice to see you. Supreme great. Cool, and Luke. G'day, friend. Hope your hands are well. I've got lots of abscission going on at the moment. So he's in senescence. So you can counter that, remember. Watch that video of mine if you want to counter it, or just let nature take its course, which is good. G'day, every day. What do you mean you can breed with abscisic acid? How does that work? Well, if you breed with abscisic acid, in other words, if you put your cultivars in with your breeding program and breed with it, it'll promote a stress and it'll activate the drought stress pathway from abscisic acid, simulating that your plants are into drought. So that will have some epigenetics code the DNA to tell the structure that it's going into drought mode, and it'll overcome that. So by you living in that environment, if you want to eliminate your cultivars always wilting, you can do that. You can add vinegar to it, to this as another cool way, because it has abscisic acid in it. I hope that explains the questions every day. Is abscisic acid same as drought stress? Abscisic acid, mate, is one of the plant hormones, and it activates drought stress. So if you put abscisic acid in around near the plants, it will pick up that it's there, and it will activate the pathway that I showed before to promote the drought stress. It'll close off the stomatas and try and save water as much as possible, simulating a drought. Hey, small tubes, how you going, mate? Hope you're dabbing really well there. It's going to warm up for you soon, they reckon. Cool, and Luke, because ethylene reverses drought stress. No, I wouldn't think so. No, I haven't seen anything to suggest that, and no, it wouldn't. It activates a different pathway. Ethylene promotes a different pathway. I don't think it would. I don't know 100%, but from my understandings, I think not. But I can't confirm that, mate. You can do the experiment and let me know. Hey, Buds and Hazards, how you going? Cool Hand Luke, or am I thinking of something else? Ethylene is also female growth hormone. That's what blocks colloidal silver. Yes, ethylene is a female. No, colloidal silver blocks the ethylene, because when it's going into its germination, it'll start and initiate. Ethylene is a female growth hormone, yes. That blocks colloidal silver, yes, because when it flips, you want to try and stop the promotion of the anthers. You want to produce the perigonal bracts with the seeds to produce the pollen. Yes, that's correct. Cool, and Luke. Do people use these hormones with PGR? A plant growth regulator has various different things, and a plant growth regulator could be these. Is these hormones? If it's called a hormone, it's produced by the plant. If you feed it to the plant, it's a plant growth regulator. So if I was going to give it ethylene, I'm giving it a plant growth regulator. But there are regulators that can cause cancer. People use some of those, and they're sold a lot in some hydroponic shops, and don't use them because they produce cancer-causing compounds that, when smoked, unlock and will pass it on through the body, because cannabis, being a bioaccumulant, it accumulates what it's given. The bad ones are either banned in the States and in Canada, and they're not banned in Australia. So please be careful, because cannabis is still pretty much illegal here. So if and you can't afford it on the medical system, I know that. So if you're scoring your street weed, you really got to be careful that people don't use PGR. Try and find out. I know it's hard asking people because you never ever get close to the grower, but it's just, you can look at the buds and you'll see, are they very, very hairy? And that's grown with components that are cancer causing most of the time. That's a good telltale sign. If you buy a pound, if you look at a big bag, and then it's got all hairs, pretty much it's all hair weed, and you look under your microscope and the trichomes are very small, only on the anthers, it's a low percent. So PGR weed is it's usually under 10%, the stuff that I've graded. Don't smoke it. Tell them no, you want proper weed with calyxes. You want the perigonal bracts, the little chunky things. You don't want it all hairy. Hey, Monty, top of the morning to you, Monty. How you going? Momentic Meds. Cheers, mate. Cool, and Luke says, I wonder if a bit of cutting powder in a medium would help root growth early. Cutting powder. Maybe striking powder, or if you look on the side of it, it'll have the rooting compound. I think you're talking about the roots for clones. You dip it in and it promotes the roots. And if you look at that, it'll have IBA, indole abscisic acid, and which is an auxin, and it should have what's the other one? Naphthalene, NP. I can't even remember it now. If we go through those slides, it'll say it. But yeah, look on the side of it, mate, and it'll have the two form IBA and what's the other one? NBA. No, AB. No, and the other one. They're both auxins. So you can put too much in. This is the problem with auxin. If you put too much in, it'll have a toxic amount and it won't give you the benefits that you want. So it's a fine line with plants. You have a deficiency amount, and then there's a normal growth amount, and then there's a toxic amount. And these hormones are talking about, mate, you need scales that go to 3 decimal places to weigh it. Is it 3? Yeah, 3 decimal places. That's 1, 2, 3. 3. So it's very, very fine amounts. So yeah, be careful. Play around with it. Thanks for the questions though, guys. This is good. I've seen purple. Cool, Luke says he's seen purple stuff that looks too hairy. Good way to get around the red look. Purple stuff. Well, maybe, oh, I'll have to get some photos. Okay. Oh, yeah, good way to get around the hair. Dave says, the worst thing about PGR is really not many people know about it. Yeah, I know, Dave. It's very unfortunate. And a few hydroponic shops are promoting it too, mate, which is very bad. So be careful of where you get your knowledge from and your growing implements from. There's one shop in Victoria I know of that sells 90% of PGR in his products because he promotes it, because he gets it cheaper from that manufacturer. And it's Psycho, so don't use Psycho. Actually, just in general, don't use Psycho because they got bought out a few months ago by Monsanto Bayer. So they've probably gone and changed it to make it even more toxic. So please don't. The reason people grow with that is it stops the stretch. So these illegal people, they can just use the PGR and they know, all right, it's not going to stretch. Well, how about get some education and learn your lights in your cultivar and manipulate your stretch better so it's not going to cause, but they're not connoisseurs. They're people who are after money, and they're the people that are not welcome on this channel. I don't promote any types of unsafe or unhealthy practices, and I'm a connoisseur and promote only those pathways that are activated to give better plant health, so it gives better smoker health. And if you haven't got the right cultivar, grow it. Breed it. Make it better for you. Manipulate it. Something along those lines. Oh, there you go. NAA, naphthalene abscisic acid. Yes, thank you. That's one of the oxidants. I'm down to the bottom. AussyCC, can you show us how to spot PGR? Yes. I'm going to pull up some. This might be pretty funny, actually. I wonder where that is. That's in. Oh, I know. You might be able to get a bit of a laugh here. Red bud, where are you? There it is. Is there any hairy buds in there? Oh, there's not. Actually, there might be. Here you go. Oh, it's a bad photo. I'm going to show you anyway. This is a bowl that has about 70 different strains back in the day. Allow. No, it's not the one I want. This one. So this is about, oh, it doesn't look like 70. Well, there was at least 50. Each bag used to put. Anyway, so in here, there should be some hairy ones. There you go. There's one. There's one too. Big, chunky, red, hairy thing. There's one there. So those are the batches. Fuck. Sorry for swearing. Jesus. I've got to watch what I say. I'm not talking to mates here. Those were, well, they come out of, they're PGR buds. There you go. The rest aren't. You can see they're nice and tight. You can see more calyx. You can't see as much hair. See the difference? Look at this one in the corner here. So you see all the perigonal bracts all the time. There's no hair. Where these ones here, they're dominated by hair. Where's another photo? No. Here's a bit of a zoom. So maybe that one. It's hard to see with this really poor photo. This was taken many years ago too. This is probably, what pixel? This would be a low pixel rate. Where do you find out the information? Anyway, it's just low. This is taken probably, this is a 15-year-old photo. All right. But, there you go. That's the best one. So yeah, that's the best way I can sage every day. That one there, definitely see the big chunky stem. But yeah, that one, that's very poor trim. They don't care though. They're all about money, mate. Whatever weighs the most. That might be a bit of PGR there. Predominantly way more hair than usual. You want it to have a little bit of normality to it. Actually, I think I've explained it enough. I hope that helps, mate. All right. when send us photos, what can you, every day says, when you get sent photos, what can we look for to know, or can you know when you smoked or vaped? Under the microscope, get yourself a portable microscope and you'll see there's not many trikes. If you can grade normal weed, I've been grading it since 2005. I got my microscope. That's about the best way to tell. And from that, you can find out various things that's going on. I found out once that through the naked eye, it looked good. Under the microscope, it looks like it was all powdered to the bench, like when you were baking. And what that person has, that grower's done, when they did a harvest, they soaked their buds in this formula that adds a little bit of sticky crystal to it and a bit of weight. So, yeah, that's a beneficial thing from having a microscope. You can see what's going on, how the grower grew stuff, and make sure they did it properly. Monsanto's, yes. Wash hands. Cool and Luke's should have some good slides, I reckon. I don't know what that means. Jay, I have a good, oh yeah. Monty has a good video on PGR, he says, on his channel. Thanks, Aussie. Oh, good. Yeah, sweet. Okay, next topic. It seems some growers are making their weed look really good too these days, and it still be PGR. No. Oh, not that I haven't played around with it for far out for decades, mate. Because now that I know what it's about, I stay far away from it. And anybody that's involved in it, I try and talk them out of it. If they're not going to be talked out of it, they can just grow themselves. And I don't want to know them because they're in it for money. They're not in it for health. So possibly, yeah. But the hair thing is a definite telltale standout. If it's way too hairy in Australia, we're back to it. Yeah, there's no structure either. Correct. Well said. It is. It's really horrible. That's why the density of the trichomes is so low. All right, back to the share where I was up to. Let's get back to some hormonal stuff. Bodily fun. Cross them off. It's shoot development. That's right. The pathways for the shoot development. So the gibberellins go and they'll halt the SVP gene and then the SOC gene will be activated. And that SOC gene in turn performs a few different pathways that gets action happening. They stimulate more genes, and that's how that cycle works of shoot development in this little bit here. I'm sure you don't want to know that way. Regulation of flower time is through a floral receptor, which is an FL1 gene, florigen. And that can be also promoted. I'm sure I talked about this. Actually, I did. I talked about this regulation of flowering time slide in that countering senescence video. It just goes on about all the different pathways again that can be activated and stopped from promotion of different types of ethylene, abscisic acids, different other florigen activating substances. Plant hormones. All right, very good. That's a real basic one. Apical meristems, yep. Cytokinins does the shoots. Gibberellins does, yep. Gone through all that sort of stuff. Is that here? No more left and right. All right. So we can go back to, so I've been through all of them. Plant growth, plant parts. I don't want to go through all of the individual ones. Do I have to? Do you just want me to? Someone talked about ethylene. And it's, see just in here, I've got about 100 slides just on ethylene. Here's the triple response. So you've got your shortened hypocotyl, and it's thicker than normal. And then if you were to pull it out of the medium, it'll have a J hook as its radical. It's pretty, like, you'll just easily be able to see it, that you've got too much ethylene in the room. They're springtails, which are very beneficial. They're on water, sitting on top of the water on a tea that I made on leachate. If you were to, the way to get them into the water is to put a water conditioner, like a yucca in, not a water conditioner, a water, you want to turn it hydroscopic. So it's hydrophobic at the moment, and it takes away the surface tension. And by adding this wetting agent, it will reduce that, and they'll go into it. More ethylene, more quiescent centers. There you go. So here's a cool test that was done, and that proved that there's more quiescent centers. And you know, if you've got your QC cells, that there's more chance that you're going to get really fast development of everything around it. So this was an experiment done, and it proved that. So you can see here, they're staying them up the top here. They're them at the start there, and it goes on. Look at them; they get a little bit bigger. So that's one thing of ethylene, promotes quiescent center and columnar cell division. Columnar cells are the ones right next to it at the tops there. Oh, CTR1 is your triple response mutant. Oh, okay. CTR1. What's that stand for? Oh, yeah. Constrictive triple response. See how all the genes, they're related. So if you want to know what the gene means, it'll be written in the name. It's like the ETO gene is the ethylene triple over expression mutant, so ETO. And the CTR is just like that. All the other genes are the same. So it makes it easier to remember. Because manipulating genes, it's like a cannabis plant. It's got 35,000 genes. And if we can manipulate every one of them, we can control exactly what we want it to do. So by learning them a lot more and how they are activated and stopped, and you can do that. Rooting from a flower stem. Yeah, that's cool. Roots from a flower stem. So that's I've seen this from a friend also in wherever he is there, and he's had the same thing come out of buds. And I would have suggested to him to get a air gas constituent test for the air, because something is going on, because it's not normal. So it would have been ethylene. So ethylene has been used for thousands of years by Egyptians to ripen figs. They do it now to ripen bananas. Ethylene is produced in accordance with seed tissue growth. No ethylene production is detected in dry state, suggested that internal concentrations of ethylene in intact seeds are near threshold required to induce germination. So no ethylene's in the dry state. First peak of ethylene production coincides with the splitting of the seed. ACC is a precursor of ethylene, and what's that stand for? And it greatly stimulates the ethylene production. Ethylene helps to overcome dormant seed with apical dominance. That's why I will always put a banana or an apple in with my dormant seeds, and it will help a lot. And I know that I've done the best I can. I've added gibberellic acid, and I've added ethylene, which is the only two hormones required to germinate a seed. And there you go. It's the best way to do it. And then you'll know if you put too much in by the triple response, too much ethylene. See how it happens. I'll kind of explain that. Overcome port dormancy, roots from stems. Endogenous ethylene is a natural regulator of root primordia formation or outgrowth that the GAST participants in auxin promote root growth. So it works with auxin to promote the growth of roots. There you go. You can root anywhere. Big hairy thing. The response requires unusual high ethylene concentrations. There you go. 10 microliters per litre. So 10 micro. So that's 10 ppm per litre. That is so low. You can virtually, that's what, 6 decimal places. So if you were to take off 3, so per gram, that's 3 decimal places. You were to put 1 in to 1 gram. So it's really, really hard playing around with hormones. So please be careful. Ethylene inhibits shoot elongation. Yes. So in the PGRs, there's many ways you can manipulate a plant, and this is just one way. So if you're getting into your week 2 to 3, and you think, I don't want a big stretch, go and buy heaps of bananas, and you won't get it because it stops shoot elongation. Ethylene effects on seedlings' height. See how here there's no ethylene? Here is 10 ppm, 20, 40, and 80 ppm. And look, all it does is just gives you that triple response. Thickening of the hypocotyl, and it's very short. And if you were to pull it out, you'd have a J hook. Inhibitory effects. I'll just stop there, see if anybody's said anything, because I've been talking for quite a while. G'day, Zaya, the Aussie. How are you? Weten up. Yes, that's what I was trying to think of, but he read my mind. Sorry. We pushed, Cool and Luke pushed a 6-foot plant over years ago, and nodes that touched the ground, rooted. That root system is called stolen roots. When the roots of a plant touch the ground and reroot themselves, yeah, stolen, and it's spelled the same way, S-T-O-L-E-N. Isn't that cool? So you can get adventitious roots, you can get tap roots. Adventitious is the ones that spread out, and that's what's actually nearly like this one. The big hairy one, and then it goes even more longer than that. Taproot in the middle with all the lateral roots outside, primary root in the center, and then the lateral roots go out, forming an adventitious pattern. Inhibitory, yeah, good. What's something interesting? Concentrations of applied ethylene as low as 0.25 nanoliters. Nano, that's milli, micro, nano, pico. So that's 1 micro. So it's 6 before. This is 9 decimal places. So if you have a little bit per litre, concentrations of ethylene of 9 decimal places will reduce growth in a cell in cannabis, medical cannabis. Fascinating. It's so low. Exogenous, so added externally, applications of ethylene or the precursor of ethylene accelerates leaf senescence, while treatment with exogenous cytokines delay it. I go over this extensively in that video I keep talking about, referring to, about cannabis senescence. It goes into ethylene is produced at the highest rate of antithesis and during early development when the auxin is developed. In virus-infected plants, stress ethylene is produced. Wow, there you go. So you don't want that. It slows the plant down and starts to finish the plant. Ethylene in flowering stage. Nevertheless, most of ethylene synthesizes in bacterial or fungal diseases is generally also plant origin. So a lot of plants, they put out a lot of these gases. They'll turn it into a methyl signal and it'll travel into the air, and then it'll be airborne. They can make it volatile, the plants. Fungal infections generally caused increased ethylene production in plants. So if you've got a fungal infection, it generally caused ethylene to produce more in your plant. So if you've got an ethylene meter, which I've hunted around for because I want to play around with it, they were quite expensive and I can't afford much. This is something that I would have found out. Actually, good time to talk about that little money button under the bottom. Thank you for the people who do ticket, and it helps me a lot. And it's just a $5 thing for if you want to subscribe per month for thanks, $5 or $8, $80 for some lectures that I haven't released, private ones. $100 is for like one-off video chat where I can help somebody do their medical cannabis things somehow, hopefully a little bit legal because I only promote legal things. And $300 is for a company or for someone going through who wants weekly video calls and they might want my professional opinion and I can help. And I'm not a doctor or anything like that, but I can give my educated opinion and help in that way. So thank you for those. Ethylene also swells nodes. I haven't seen that. Inhibition of ethylene of its nodes and stems. it'll shorten them. It'll make it grow stunted. Induced horizontal growth of stem against gravity. Induction of horizontal growth against gravity. Evolution of ethylene from plant tissues can be detected by certain biological tests. Did that before. Preparation of standard sets of peas. Oh, that was just explaining the one previous. Epinasty is relaxing behavior seen during flooding of plants. Okay, we don't want that. Yeah. So sometimes you might get this type of a wilting effect, and that will be too much ethylene. Ethylene definitely causes this type of growth patterns in plants. So if you're getting obscure growths, it'll be producing the ETR1 gene? Was that it before? No, I did not quite that. Ethylene-induced fruit ripening. Yes, that just works. This is it's a very simple way. It doesn't tell you any pathways, it just tells you it does. All right, well, it does. It actually does too. I've explained that a lot. Oh yeah, here's a bit of another triple response. So I see the J-type hook in its root, and then the shortening and a thickening of the hypocotyl. Perfect. And it's the end, is it? Oh, it is. Okay, that's a good way to finish it. Ethylene. Can we mark this one ethylene? Banana farmer, how you going, mate? Green apple for ethylene down the center of your pineapple to help induce flowering. Wow, many different techniques to do, many ways to manipulate the plant. It's cool. And if you want to look at the ethylene, the amount of ethylene produced by ripening fruit, if you cut it, you get more ethylene production. I've never really had to cut mine. I'll get 1 or 2 apples in with half a dozen seeds or a dozen seeds, whatever I'm dormancy breaking, and that'll be enough for me. So I'll put it on this little shelf that I made and put it in a bag so the ethylene stays high and it works perfect. Great trick. Speaking of tricks, nice trick, Nana. The plants just being near them. Yes, it has to be, it can be up. If it's upwind, it won't get any responses if it's in unless it's in a sealed room. But if it's downwind, it's perfect. I've proven that by some breeding practices I did a few years ago where I was transferring some genes from plants, and you can see it transfer through plants. I took some photos of it. It's very cool. That was when I was trying to give fungal resistance, I think, something along those lines. So I had to, anyway, back to the point. The plant's just being near that. Yes, that's true, mate. Within, that's why they don't want the leaves to touch, because every problem with that next plant will go through that other plant touching, and they don't like it. They've also got calcium signals in there that get activated in there when they get touched. So that's not good for growth production, for it just to grow out, because they'll grow around it when they don't like it. But like us, we don't like being touched. Hey, Kemet, how are you, Kemet? Right at the end, Kemet, just about to finish, buddy. It was an open topic, but it's changed into hormones, and it's more so ethylene I've gone on about today. An hour about ethylene. So is there anything else anybody wants to explain about anything? Look at this little slide here. It's supposed to be a healthy show. Yeah, health benefits. All right. We got any ethylene things? Peoples, there's, what else can I tell you about ethylene? Let's go back into the thing. Ethylene. About this one. Didn't show this one. All right, I'll show you more about ethylene. Still got a few minutes to go. Most questions aren't really getting pounded. Share screen just to finish off on the ethylene topic so I can make it nice and clear. Thorough, more words I was looking for. This is ethylene-induced senescence. So it would have activated that, and you can see it there in the red. So the Indole abscisic acid gets produced at the shoots. It's a polar transport and goes from the shoots down to the roots. And then the ethylene kicks in and it starts the C2H4, which is ethylene. That's what it looks like. Senescence. It early ripens. Ethylene in smoke has long been a practice to ripen fruit. This practice has included ripening pears in a smoke from incense, or gashing of unpollinated figs has been practiced. The ethylene produced upon wounding induces ripening. Yes, that was what, this is an old practice, so it's nothing new that I've done. It's old school agriculture that we can still use in our medicinal cannabis. And ethylene and smoke. Well, if you use kerosene is another cool little tip. Kerosene off gases and produces the C2H4, the ethylene. So a lot of growers many years ago who used to heat in North America with kerosene wondered why their crops finished early. That's the reason why. Another way to finish them early. Cousins in 1910 showed oranges shipped with bananas caused the bananas to ripen prematurely. Later work showed it was the few damaged and mouldy oranges that actually produced the ethylene. Yes, but I don't promote mouldy oranges or anything mouldy sitting in you unless you're a breeder. You don't want to use these practices because they can be dangerous. And the mycotoxins produced by them, depends what variety, it could be very harmful to your body. Be careful. These only should be done in lab conditions. I tend to, when I breed or do things like this, I'll do it behind closed doors. It'll be where I'll be involved in tissue culture stuff. I want to maintain all the good safe practices. In 1901, ethylene was identified as a compound that affects plant growth by Dmitry Neljubov in the big motherland up there in the Northern Hemisphere. He traced the source of the strange growth patterns to his pea seedlings and found that they produced ethylene from his kerosene lamp. There you go, we've gone through that. Even trees near street lamps showed higher leaf senescence back in the day from the gases that they burned inside of it. So this leaf that's planted here would ripen faster than the one that's not near it. In 1934, again, purified ethylene from ripening apples, demonstrating that it is the exogenous hormone. So by the adding of it externally, it works. In 1959, a gas chromometry. So they did a test in a lab with a gas chromometer, and they found that it was the ethylene that induced the signals. Cool. And the gas chromometer revealed that ethylene is a cause, not consequence of ripening. So see on, this is a rad chart. See on the chart here, you can see after 2 days, it just spiked. The ethylene production really went high and associated with CO2 production, which is a thick black line. I've proved this from ripening cannabis lots because I use CO2 meters, and I know when my CO2 meter, it's like this. Depends how fast it dries, of course, but it'll stay high for very high for 2 to 3 days, for maybe 5, 6 days, CO2 production. So the cells are still alive and they're still respiring. So they'll produce off gas a lot of CO2, and they'll also produce a high spike of ethylene. Only for a short period though. See, it's only once their cells are still alive in their finishing state. The air oxygen is required for ethylene production as well. So if it's not mixed with the right amount of air and nitrogen in the air, that the ethylene won't be produced. So you want to get, that's why the bag technique works well, because it's a good blend. This ethylene precursor was called compound X. ACC is a precursor. Compound, right, stop. There you go. Compound X is ACC, aminocyclopropane carboxylic acid. So if you can get this precursor, like tryptophan is a precursor for auxin. So I use tryptophan from another source because I'll almost tell you, spirulina what's the name of it? Spirulina athroplatensis is the bacteria I cultivate, which is 70% tryptophan. So I got an organic source of tryptophan, which is a precursor for auxin. This is another way. So you can go and you get your aminocyclopropane carbolic acid, and it'll also just be that only be one pathway needed to turn it into ethylene for the plants. And here's the synthesis, how it breaks down, and it goes through its processes, which I'm not going to go through. As you can see in there, they're very detailed. Mutants, and the ETO1 mutant, which is ethylene can't remember what it was, but the ETO gene mutants, that's a dominant, has triple response phenotype. So ETO1 dominant gene produces a triple response. Okay, so that's what I get a lot, but it's only because I've induced ethylene. It'll grow out. ETO1 interacts with AC55 gene and negatively regulates the ACS function. Thus, the gene mutants have high ethylene content. Mutants are fun. This is more of a breeding thing. I'm going to skip past that. Ethylene oxidization. This is its pathway. How ethylene is a very flammable gas, so be careful. So if you get too much of it, it'll it can react very heavily. So this is how it just breaks down. It has the oxygen and it starts to break down into glycol, into the acid, and then to the carbon dioxide when it's fully separated. There's a fruit ripening from back in the day. Oh yeah, this is a technique. This is that room I was talking about earlier. Right. I wonder why didn't you start here? Anyway, so you can see how they pump ethylene in. It's spikes it up to, it's very high too. It's, I'm not going to quote a number actually. It's very high. And it ripens it. That's how they do it. Ripening on the road, yep, they've just got trucks. I like cartoons. They've got trucks that do it. They probably don't look quite like this, but I reckon they do. They come into the lab, look, they got all these things into the fruit shop. It's cool. Fruit ripening. Yes, it just does work. Silver inhibits. This is what you were talking about before. I think Coolhan Luke, when you were saying about the ethylene blocks the colloidal silver activities, and you're true. And this is a slide showing backing that up. So the silver thiosulfate is the same thing, SDS, colloidal silver. You can use another silver particle. I can't remember it now, but that's what it does. It just blocks it. So the ethylene just blocks it. Those particles that we'd put on if we want to try and induce and reverse the plant, it blocks it. Summary so far. Ethylene promotes senescence. Ethylene ripens fruit and further promotes respiration. Ethylene was identified as an agent in bearing, that's ethylene affects seedling growth and its biosynthesis, its oxidation. Oh, righto. Ethylene's, I think I covered enough. Oh, there you go, look. The thickening of its triple response, thickening of hypocotyl, shortening of the root, and it gives you the J hook. There you go. This is CTR1, that's what I was thinking of. The constituent for triple response mutant. So it's activated the CTR1 gene heavily when you get that effect. CTR1, CT-tripleine, constitutive triple response 1. CTR1. Right, I'm just trying to learn as we're on the fly here. Here you can hear my learning techniques. Morphological features of the triple response. We're still going on a fair bit about that. When germinating in the dark, oh, here we go. It'll tell you about the ethylated. When germinating in the dark, impeded seedlings produce ethylene, which confers the triple response. It also helps them get round things by turning, because see how they're turning and twisting? That helps them get round stuff. So as a rock here, it'll eventually get round because the ethylene's built up in here, and it does that. So by treating dark-grown seedlings with exogenous ethylene response mutants could be identified quickly. The response of ethylene is very rapid. Yes, within 30 minutes or so, you can really see it, because it's it goes in through the plant. It's got a few pathways where it can take things in, like the stomata and that dermal layers of the tissue. And there's the other one, and through the other ways that it can absorb them. So it just goes in very fast through its respiration. Ethylene signaling mutants. The EIN, ethylene insensitive mutant, shows no response to tripling effect. Wow. So you can even breed that into your breeding program if you want. So ethylene, it's insensitive mutant, which I would hate to have that, because I love using ethylene for dormancy breaking. So that might be another practice. If you tried to dormancy break your medical cannabis seeds and it hasn't worked, it could be an ethylene sensitive mutant. Ethylene signaling pathways, yes. It's ethylene blocks the receptor, ETR1, which produces the kinase, which break down, and it blocks the EIN2, which has its transcription factors in its RNA, and its EIN3, and its ethylene response factor 1, and then it goes into its target genes. That's its ethylene signaling pathway. Here's another one. I don't want to explain all that. I think it's been going on long enough. ETR1, yep, it binds, very good. ETR1 mutation. How can a mutant receptor have a dominant phenotype? This is getting in breeding. How can a mutant receptor have a dominant phenotype? By bringing it into its DNA. The receptors negatively regulate the response. A receptor that always shuts off signaling is dominant. Yep, that's how you tell with that. Also, function mutation in ethylene receptors show constitutive ethylene response. That's the CTR1, ethylene triple response. CTR1, ethylene response. Ethylene. Anyway, that's ethylene receptors, two-component kinase system, the cytokines combining with ethylene, reacts that way. So you get your adenine triphosphate to your adenine diphosphate. Cell currency. All right, that's enough. This is getting into breeding. Anyone still here? Oh, it's now 10. God, I go on about. Oh, Jesus. Thanks, Aussie. Zaya, yep. Where I was up to. Just going to read this. So put your questions out now, because otherwise we're going to wrap it up. I thought it was bananas. I thought it was bananas that make ethylene. You can go to a volatile organic compound, release factor of ethylene, and you'll see that it's bananas, apples, pears, and then it drops under that. So they're the things that I would recommend. G'day FM, how you going? Hope you're still hitting the volcano. I'm bloody oath I am, mate. Love the volcano. I'm going to hit it right after this. Say the Aussie, yes. FM, all right, going down. It's a hydrocarbon, same as petrol? Is a hydrocarbon same? Yes. Yep, it's in the same yucky, smelly, volatile organic compound family that reacts, as far as I know.
not very into chemistry though, mate. Well, here we go. Be careful, Banana says, banana farmer. Be careful with banana skins because they spray 2,4-D on bananas to help set fruit and ripen. Wow. 2,4-D is a bad chemical, a pesticide that you don't want. Wow. Yeah, I always use apples. I'd always suggest apples. I wouldn't use bananas. Anyway, bananas are too big. Apples are really good. And they're small. They last a long time, the apples. I can get easily 2 or 3 weeks out of it. I'll put it back in the fridge, and I might be able to do another round, maybe in another few weeks' time. So I'd suggest apples. Good. Thank you for the safety tip, Banana Farmer. I love the safety things. Keep coming, mate. Not always. I never did, but always on an option. Okay, don't know what that means. FM says, so ripening fruit makes ethylene, which makes the fruit ripen quicker. Yes. Yes, ethylene is from those pathways earlier. It activates the cell senescence and it finishes them. So by having that in the air, that's what the plant naturally thinks is going on. So if it's already in the air, the plant just goes and takes up that pathway and finishes. Goes into senescence. Having coffee. Very good. What causes dormancy? Many factors cause dormancy, mate. Seeds naturally have a dormant nature until they're put in the correct environment to germinate. That's how they're developed themselves, because they don't want to be in the Arctic and then, you know, they're going to die all of a sudden. They don't want to be in too wet and they're not going to be able to strike a root and get oxygen there. So it's dormancy is a natural factor, and age induces it as well. It helps it, promotes it. It oxidizes the tester, the outsides of the seeds, and it creates a barrier there. So the longer the time, the harder it is. Seed dormancy is what causes it. I hope that's explained it. Cool hand, Luke. Brief touch on it. On down the bottom. FM, how do some cannabis strains offer pain relief and others don't? That's to do with the terpenes and flavonoids that are in there, mate, and the combination of cannabinoids. So the ones that have myrcene in it are high in pain, and choreophyllene, high in pain. So you'd sort out those ones. You'd want ones that are high in CBD as well as THC, and that would also help with the RSO effects. And then you get your cultivars like your PGR that they don't know what's in them, that they have so weak stuff that the trichome development is so small that it doesn't hold many things. It doesn't have idioblasts because they're all just shrunk away in the plants. So it doesn't store compounds inside of it. Back to the question. How do you know some strains offer pain relief and others don't? How can you tell? What you do is you smoke them and see what agrees with your body. And that's my best way, because there's a lot of information out there. Not many people are into the breakdown of the secondary metabolites, which is the flavonoids and the terpenes and their entourage effect, which is a combination of them all put together. So this is why people struggle, because it's not a lavender plant which just has linalool. It has many hundreds of terpenes in there, and everyone has a different effect. So I can only suggest, you smoke it. If it agrees with you, grow it. That's what I did. I went to Canada, spent many thousands in their dispensaries, seeing which ones agree with my body and sort out those genetics. And that's what I've got. A lot of genetics like that, which I agree with indica dominant varieties, not sativa. And then out of that, I'll pull genes, what I like out of other cultivars, and put them into the others. And then I'm also, that's a breeding thing, getting into that. How do some cannabis strains offer pain relief? I hope that helps, FM. I always feel banana skins to my worms. Banana farmer, yeah, man, same. They do. They have a worm bin just for flowers, fruit and veg scrap.
Okay, I'm not sure what that means. OFM says, I know beta-caryophyllene is a cannabinoid, but caryophyllene is a sesquit. No, beta-caryophyllene is a terpene, mate. Yeah, beta-caryophyllene, and I don't know if it's a sesquiterpene terpene. I'm not going to pull up that chart because we've been going for a while now. Cheers for that, CC, as it's easy to get seeds legally in Canada where you are. Yeah, you're lucky, fella. Yes, that's the best thing I do, mate, is go to the dispensary, see what agrees with you, and sort those genetics. Be careful on, I'll have to tell you, I suppose. I was in a mate's shop, and these other fellas were in here. They own dispensaries in the prairie land. You'll be laughing. Or inland, many hundreds, in the middle of Canada. And they had changed Death Bubba's name to something else. And I said to them, why? I said, it's about medicine. We want to learn what terpenes, what profiles it's got, this type of stuff. And they said, because it's to do with money. People want new names. So they change the name and people are happy. They don't know the difference. So this is the problem, mate. Too many people are in it for the money and not into it for the medicine. So FM, you're really going to struggle unless you really hook up with some cool people, similar to myself, who really is into it for the medicine only. And then you'll, probably get to some areas that you want. Just go and get the seeds that you want. And even if it's got a different name and if it agrees with your body, cool. It agrees with your body's number one. Great question. Yeah, because it's really hard to know what agrees with you, because usually it's just cop that. No. Where this makes me feel anxious because I'm caffeine sensitive, so I might feel uneasy from sativa dominant varieties, which I do. It's something that's you've got to use as medicine and use it the right, the martial artist. No way. How you going, mate? How you going, martial artist? That's the way. We're just wrapping up on a big hormone discussion on ethylene, and now we're just ending on a bit of terpenes. Martial artist. So if there's any questions, or actually any questions for anybody, please drop them now, because otherwise I'll wrap it up in a couple of minutes. So you can go for an hour 17. I'll wrap it up in a few minutes. So drop your questions. And FM, maybe save you a question for next week about your terpenes, and I can go into detail about the terpene profiles and their isoprene rankings from their carbon chains. Like you said, sesquiterpene terpenes, and that has 4 carbon chains onto it, and it starts off as monoterpenes, then sequiterpenes, and it goes on like that as it goes up in rank. And they do different things because it's the length of their carbon, amount of carbon molecules that it has into it, and that's how it gives it that ranking. And there was some cool charts I've never shown before on some terpene profiles on what terpenes are good for your body. So in other words, I have epilepsy, so I want antispasmodic or anticonvulsive terpenes, and then I want that in my breeding program. So that's what I try and do as well. So terpenes mean a lot of things, terpenes and flavonoids. Cool Hand Luke says, I want a strain that I know a bit of history about. Yes, mate, like Death Bubba. Yes. Soon I'll be able to give out some of my genetics when Australia goes legal, and then you'll know some history, and then you can see what works with you. So it'll be cool. Yes, that will be cool. I gave away many. Actually, I'm not going to talk about that. Cool, martial artist. Very cool. Thanks. Just found your channel. I didn't know you were doing this. I'll be here more. Yeah, it's hard to. I don't promote myself, mate, and most people don't promote me. But thank you, and it's great to see you again. FM says, thanks, AussyCC. Cannabis is still an illicit drug, but is also a commodity with profit margins. Yes. It's horrible, mate, isn't it? Needs to be, more connoisseurs like myself, and hopefully quite a few of you out there are connoisseurs too, so we can really fine tune and get the best out of our cultivars. And if that's not good enough, we're going to manipulate it and change it through hormones or through breeding programs to make it way more beneficial for our bodies. It's awesome fun. Cheers, cool, and Luke. This time every week, Marshall. Yes, it is, mate. In 7 days' time, I just do the one show a week. And I used to go through some of my lives. I used to put really good titles into it, do a fair bit of research and stuff like that, and give the latest news on medical cannabis. And I haven't for about 3 or 4 months. I will in a few months' time, but I'm just still very busy with the projects that I'm doing at the moment. Sorry. That's why I'm doing these open streams. So it's just usually like this stream here, which is an open medical cannabis topic where just bring you questions. And then we can start. Like today was hormones. So I'm going to retitle it Hormones. Vinny, cheers. Montana, here you go, Montana. FM, make sure to drop a like. Oh, yeah, thanks, FM. Ziggy D, legalisation isn't far away. Hey, it should be at the end of the year, mate. The way that things are going, yeah, it should be the end of the year. It might be earlier for Victoria and Tasmania, so I might be able to pass some seeds from ACT to those areas sooner, but I've got to do it all legally because I want to respect the law. Don't want any problems. Spend enough millions and all of a sudden it's legal. Yeah, the government's got to make their cash cow. They've got to cash in first, mate. But it'll happen, I hope, so us poor people can still have our good medicine, better medicine, because post-harvest productions of medical cannabis isn't spoken about enough. I did a course on that last semester, and it really opened my eyes to all the different things that can happen. So the people can grow it fine. They've really got that down pat, but post-harvest productions, they lapse a bit on. Far out. Ziggy D, FM. Oh, he's at Ziggy. Sure, bro, no way. How you going, sure, bro? Woohoo! Nice to see you, brother. Yes, sure is. Oh, what? Mace, Big Will. Yeah, how you going, Big Will? That's it. I'm smiling. I can see you. Good on you, mate. Today was a discussion on hormones, plant hormones. Went into it pretty detailed on ethylene. So I didn't discuss the other 9 plant hormones. Nice to see you though, brother. Ziggy D. Yes, I had a pretty in-depth conversation with Nexus about it last night. Okay. FM, there are 0 topical THC containing medicinal products I'm aware of. 0 topical. Oh, yeah. Yeah, there's over in BC, I used to get a fair bit, mate. There's all sorts of gels and balms and soothes, and there's butter. There's quite a lot of stuff that they've got out there. Maybe you must be in a province. You're in one of those illegal, or the province that doesn't promote it over in the East Coast. Sorry to hear that, mate. You might get online because in Canada it's legal to ship through the mail, and go some BC places. There's some rad stuff place out there. Gear changer, nice to see you. All these people come in right at the end. How do you turn Mullimatic dust into dabs? Mullimatic. How do you turn your keef powder into dabs? You have to add this chemical, which is someone help me out in Canada. It's don't use coconut, don't use MCT oil. You can use that, but don't use it. It's not as good. There's another oil that you use. You add it with, and it just turns it into it, and then it makes it liquid and it stays liquid. A lot of these other forms, they hold it differently, so it won't burn properly. Where this, I'll find out. I've got to find out. There's one bloke in Canada that still does that. Actually, Main Green does too. Yeah. And also, I bought a kit a while ago that has the liquid in it. It was $90, and it was about, it wasn't 100 mils or something like that. And yeah, So there's a kit that you can get it. All it is one liquid. You just squeeze it in and mix it with it, and it blends it up. And Ziggy D said alcohol. No, don't use alcohol. It's a non-alcoholic substance, so it'll burn better. You don't want to burn alcohol. If you put alcohol with it, you've got to extract all the alcohol out of it first before it's safe to induce, in my eyes. Press it like a block of hash. Yeah, that forms rosin. He's looking to put it into liquid form. Wish they were here. They're not here. It's it's like MCT oil. You can use MCT oil. I got that ages ago, but it'll bind up your device a lot, and your filters in your e-cig thing won't be happy. Where if you use this other oil, it makes it more cleaner, and you can use it, just refill, and just keeps powering on. That was a good question though, mate. Thank you. No dabs. Yes, dabs are fun. They're really good. Ziggy loves it. Ziggy likes his alcohol. That's why he uses alcohol. See, that's fair enough. Everyone's got their own little likes and differences. I promote the healthy way though. You've got to purge it. If you can do it in alcohol or butane and stuff like that, but you have to put it in an evacuation little round chamber thing and purge it for 2 days at 25 atmospheres to make it up to standard. So it's all really nicely gone. Yes. Hour and a half. All right, this is it. I haven't heard any rag questions. Not any rag. Any questions hasn't come through. I think it's been pretty good. 7 days time. I'm going to try and find out, and if someone else can research what that device is, not the MCT oil, the other oil that you put with the keef powder, and it dilutes it and makes it vapable. I can't remember. Have a look online too, mate, for that. $90 I paid. I already said that. Okay. Spring grape. Everybody's saying hello to each other, which is very nice. It's good to have a good community where everybody's polite and nice to each other, and I appreciate that. I don't like drama. I don't get involved in it, and I stay away from it. So I really respect everybody for keeping nice and starting to talk to each other in the chat too. That's cool. Keep up the good work, all you nice people out there. Have you ever tried making hash with dry ice and mechanical screens? I've tried mechanical screens, mate. Don't do it because too much green leaf material gets into it. Put a microscope under it at the end and you'll see its efficiency is low. Where if you use water, it's very high efficiency. The amount of green leaf material you've got in the end is so low. But I've never tried it with dry ice. But I have tried it with mechanical screens. And from that will be the answer though. It'll be of a lower quality to making it with bubble hash. Anyway, where you are in Canada, between 1 Celsius and 3 degrees Celsius, trichomes separate from green leaf material. So you can nearly just leave it outside, give it a few taps, leave it in the back of your car, drive down the store, come back, and there'll be a big pile of orange-looking keef powder in the bottom, mate. It's the worst thing about North America up there, transporting it. Oh, good on you, mate. Respect. Love your show. Good job. Thanks, Sunshine. Thank you, Vinny. Cheers, peace and love to everybody. Thanks, FM. Good on you. I appreciate it. Appreciate the nice words and suggestions and all your questions too. Okay, this is 1.5. This is one of my longer ones. Wow, hormones. I must have had some hormones. It's pepped me up a bit, kept me going for too long. Unreal. No worries, gear. Mace. What is the current world's strongest strain? The world's current strongest strain is, they're up about 50%, but you can't trust some of the labs now. They're up about 40% THC, and they've got some with 10% THC, so up about 50% total cannabinoids. They've also got some with about 30% THC and 20% CBD, so it's about 50% total cannabinoids. I play around with 25 to 30% total cannabinoids, and some of them go into the high 30s, the 1-to-1s and 3-to-1s that I got. But it's mostly in the mid-20s to high 20s THC that I like, for me. Thanks for the question, Mace. Ziggy, phones nearly died. Yep, CC, I'm going too, mate. Ziggy D's out, so I'm out. Choose CC, talk to you again. Yep, good on you. Everybody's bailing. Oh, so am I. Thank you. Happy breeding, happy growing, and good health to you. See you all in 7 days' time. Bye-bye.